All right, let's dive into one of biology's most fascinating puzzles. We're talking about the C-value paradox, a perplexing observation that the total amount of DNA in an organism, its C-value, often fails to correlate with its perceived biological complexity. This paradox is dramatically demonstrated by organisms like the single-celled amoeba, which can possess far more DNA than humans, and by closely related species showing enormous differences in genome size.
So this is the core enigma right here. You'd think, pretty logically, that a more complex organism, like us, would need a bigger, more detailed genetic instruction manual. But nature, well, it just doesn't seem to play by those rules. And that's exactly what makes this whole thing so compelling. It completely challenged what biologists thought they knew about how genomes are put together.
And we're not just talking about some rare one-off exception here. The scale of this paradox is just staggering. We see these huge, totally inexplicable differences, not just between, say, a simple organism and a complex one, but even between species that are basically evolutionary cousins, living almost identical lifestyles. Okay, so let's get into some of the specific examples that really show how perplexing this genomic puzzle is. I mean, these are the cases that had scientists scratching their heads for decades. So first up, the single-celled amoeba. I mean, this thing is essentially a microscopic blob. It's got no brain, no organs, no complex behaviors to speak of. And yet, its genome is absolutely monstrous. We're talking about more than 10 billion base pairs. Those are the individual letters of the genetic code. To put that in perspective, our human genome is around 3 billion. It's just wild. This next one really brings the paradox home, especially between close relatives. So we've got two species of shovelbugs here. They live in the same kind of environment, and they split from each other only about 5 to 10 million years ago. That's a similar time frame to the human-chimp split. But get this, the Canadian species has a genome of 14 billion base pairs. Its cousin down in Texas, only 3 billion. That is a 10 billion base pair difference for two creatures that are, for all intents and purposes, incredibly similar. And this puzzle doesn't stop with animals. It's huge in the plant kingdom, too. This is what's famously known as the onion test. If you look at the group of plants that includes onions, their genome sizes are all over the place. We're talking from 7 billion all the way up to over 31 billion base pairs. That means one species of onion can have 5 times more DNA than another just to do the exact same job of, well, being an onion. So all this extra DNA, what's it even for? Well, the resolution to this whole paradox finally came from a pretty revolutionary and at the time, kind of controversial idea, the junk DNA hypothesis. And here is the formal definition. Junk DNA is defined as non-functional genomic elements, such as transposons, pseudogenes, and repetitive elements that function as genomic parasites. They lack a selected effect function, meaning they can degrade, be deleted, or shuffled without causing a negative phenotypic effect on the organism. So the basic idea is that a huge chunk of the genome isn't actually doing anything functional for the organism. It's almost like baggage, bits of selfish code that just copy themselves and build up over evolutionary time. And this extra stuff, this junk, could finally explain those massive differences in genome size. Now, an idea this big doesn't just pop up out of nowhere. The concept of junk, or non-functional DNA, was actually built on decades of theoretical work, way back in the field of population genetics, long before we even had the technology to sequence entire genomes. It really all goes back to this concept called mutation load. Let me break it down. Imagine every single important functional gene is a critical part of a machine. Well, the more critical parts you have, the more chances there are for something to break, right? In genetics, those breaks are harmful mutations. So scientists figured that if an organism's entire genome was made up of nothing but critical functional genes, the sheer number of harmful mutations would just be too much to handle. It would eventually drive the species to extinction. There had to be some kind of upper limit. And you can really see how this idea built up over time. It starts back in 1937 with J.B.S. Haldane laying the groundwork with his theory of genetic load. Then, flash forward to 1950, and Hermann Muller takes that idea and argues that because of this mutation pressure, humans probably can't have more than about 30,000 functional genes. And finally, in 1972, Susumu Ono is the one who really ties it all together. He's the one who formalizes and popularizes the term junk DNA to describe all that non-functional stuff he predicted was filling up our genomes. But look, this was all still just theory, right? A great idea, but the junk DNA hypothesis needed some actual physical proof. And that proof finally arrived in the late 1960s, thanks to a really clever technique called C0T analysis, or just C0T analysis for short.
So here's how it works. You basically take double-stranded DNA, heat it up to pull the two strands apart, and then you cool it down and measure how fast the complementary strands find each other again. The core idea is actually super elegant. Imagine you're in a huge room full of people, and everyone has to find their identical twin. If there's only one set of twins in the whole room, it's gonna take them a while to find each other. But if the room is filled with hundreds of sets of twins, they're all gonna pair up way faster. COT analysis is basically doing that with DNA strands.
And the results they got were a total breakthrough. When they ran this test on simple organisms like bacteria, what we call prokaryotes, the DNA strands reconnected pretty slowly. That told them most of the DNA was unique, single-copy stuff. But then, when they tested more complex life like us vertebrates, eukaryotes, huge chunks of the DNA snapped back together almost instantly. This could only mean one thing. Those parts of the DNA had to exist in thousands, maybe even millions of copies. So the interpretation here was crystal clear. The DNA sequences that paired up super fast, they had to be highly repetitive. I mean, they had tons of copies, so it was easy to find a partner. On the other hand, the unique sequences, which include most of our critical protein coding genes, they took way longer to find their one and only match in the mix. This was it. This was the first real, concrete, physical evidence that the genomes of complex organisms were just packed with enormous amounts of repetitive DNA. And that brings us to the grand conclusion, the beautiful solution to the puzzle. If this repetitive DNA constituted the non-functional junk, it beautifully explained the observation that some organisms possessed vast amounts of DNA that did not correspond to increased organismal complexity. While molecular techniques like DNA-RNA hybridization also supported the notion that only a small fraction of the genome produced proteins, COPT analysis demonstrated the massive proportion of repetitive elements that were likely non-functional.